In this project, we used multiple materials to help us power through this project. We had gloves that help us protect our hands and to prevent any contamination. We had plastic tubes and tube covers to help us make the fly habitat. We had white cotton balls to allow us to prevent the flies from leaving their habitat, and we also had fly food and deionized water, which we mixed together to make their food source that they could live off of. We also had tape to help us distinguish both tubes during the project. We used ice as a natural anesthetizing agent so that we could view the phenotypes of the flies under the dissecting microscopes every single week.
In this experiment, we started off by preparing 2 vials for the fruit flies. In the next step, we placed the vials with flies into snow to anesthetize the flies. After the flies fell down, we placed them on a weight belt, which was placed on the snow so that the flies can stay down. We then selected 5 wild-type females and 5 white-eye females and placed them in vial number 1. Then we selected 1 wild-type female and 1 white-eye female and placed them in vial number 2. We recorded the data in our lab notebooks, and each week we anesthetized the flies to record the phenotype ratios, and then we released.
The only deviation we made from the protocol is that one tube had more food than the other. But the main issue we had was one fly from the 1-to-1 ratio tube died off early, and we had to replace it with another wild-type female fly. For the future considerations, we want to improve on our techniques in terms of phenotype characteristic and anesthetizing the flies, and we also want to keep a consistent environment next time. We want to make sure that the flies are in a consistent temperature without being too cold or being too hot.